Hey everybody, welcome to Molecular Diagnostics this semester. I just want to give you a quick little video and give you one major hint in the way I teach. I tend to repeat things that I think are incredibly important. So if you're listening to the lectures and you're hearing me repeat certain things, I would take note of that if I were you, because it usually means that there's going to be at least one question on the midterm and the final exam on that concept. And it's highly likely that there'll be a question on the quiz also. Another thing I want to go over is a few things about DNA. Now, all of you have learned about DNA, all of you know DNA, but if you haven't had a molecular biology course or a cellular biology course for quite some time, you might be a little bit rusty. So things you definitely want to make sure you know are the fact that DNA is double-stranded, of course, and the fact that it is anti-parallel. So anti-parallel is one strand of the double-stranded DNA is 5' to 3', and the other strand is 3' to 5'. And DNA is always made by adding another nucleotide to the 3' end. So it runs 5' to 3'. You also know your nucleotides. You have your G, your A, your T, and your C. And G always binds to C, A always binds to T. So when you're given a problem on a quiz or an exam and you're given a string of nucleotides, which is one strand of the DNA, you should be able to say what the complementary strand is. Now, I want you to recall, though, that DNA is anti-parallel, and that means that you have to really pay attention to what direction the DNA is going in. So if on a quiz or an exam, I have something like G, A, T, C, C, A, G, and I'm asking what the complementary strand of that is, I might put something that might be a little bit confusing, where I will put the actual nucleotides that are complementary. So G binds with C, A with T, T with A, G, G, T, C. You might see that, and it is the right complementary sequence, and be very apt to choose that. But in fact, since it's the opposite strand, it would have to be this. It would have to be the 3 to 5'. So that's just one thing to keep in mind when you're taking quizzes and exams in this course. What's complementary and what direction is it in? And remember that DNA is double-stranded, RNA is single-stranded, and you should know what nucleotides to expect in DNA and RNA, which are purines, which are pyrimidines, which have triple hydrogen bonds, which have double hydrogen bonds, and all of those things that are in the lecture that I might repeat several times. Just know those for the quiz and keep them in the back of your mind. Study, get everything in there, because you'll see everything on the midterm exam. Good luck in this course, and don't forget, post questions on the discussion board whenever something is not clear or there's a concept that you're not understanding. Because the thing about molecular biology or molecular diagnostics, the more you understand the theories, the better off you're going to be. A lot of CLS courses like microbiology, hematology, they require memorization. Now, molecular diagnostics is a little different. There's some memorization, there are some definitions you have to memorize, but the more you can actually understand concepts and apply these concepts, the better off you're going to be. So, any questions you have, send them my way. Good luck.